Hello, everyone, and welcome to this webinar for the LabRoots Genetics Virtual Week. This is Angshumoy Roy. I'm an associate professor of pathology and pediatrics at Baylor College of Medicine, and I also serve as the medical director of cancer genomics at Texas Children's Hospital. The title of my talk today is Laboratory Implementation of Clinical Genomics in Children, and I have no disclosures to make. So let me start with the objectives of this webinar today. If you're joining this webinar, I would expect that you would have, after this session, a good understanding of the different mutation landscape in childhood cancers as compared to adult cancers. I would focus on describing the structural variations, primarily fusion genes that drive different childhood cancers. And I would show some examples of the clinical implementation of genomics in children and how these can lead to broad utilization of these technologies in the clinic and provide useful biomarkers for cancer management. So let me start with this one introduction slide that came out of a pan-cancer study in 2018. These are landmark publications in childhood cancer by Grosvenor et al. In Nature, back-to-back publications showing the different landscape of childhood cancers in adult cancers. At the top over here, what you see is a plot of the number of coding mutations in childhood cancers in compared to adult cancers. So on the left, you see pediatric cancers. They have a horizontal purple bar indicating the pediatric cancers. And to the right of it, you see some of the adult cancers marked with the green horizontal bar. And I think you can appreciate on the y-axis the number of mutations by different types of tumors have been plotted. And you can appreciate by looking at that in general, pediatric cancers have a much lower rate of coding mutations compared to adult cancers, which have a higher rate of coding mutations. Now, this is something that has been known for a long time. And so this paper just merely emphasizing that this is true and significantly lower in most of the pediatric cancers that were studied. Now, having said that, you can see also towards the right of that horizontal purple bar that some of these cancers actually can have very high rates of mutation, sometimes even as high as adult cancers to the right. And so these are often referred to as pediatric high or hypermutated, or even ultra-hypermutated, depending on the total number of mutations. Now, if you look at the bottom, you can see the fact that the number of structural variations in childhood cancers. So on the left, you see the number of structural variations by different pediatric cancer type, and you can see that some of these on the right again do have a high number of structural variations in their tumors, and so adrenal cortical carcinoma and osteosarcoma. And to the right of the lower panel, you can see a distribution plot of the type of different mutation classes in these tumors. So in green are cancers that are driven by mutations. These are small nucleotide variations or indels, whereas the purple highlights the structural variation, or in this case, copy number or other structural variations, including translocations that drive different types of tumors. And you can see that there is a broad category. There are some tumors that have both green and purple in those bars, some tumors that are completely driven by green or mutations, and there are some tumors that are predominantly or completely driven by structural variation. And so you can see that structural variation plays a fairly important role relative to mutations in pediatric cancers. So that's one important take-home message from this. And that also informs how we design genomic diagnostics in children's cancer. So in this rational design, it's very important to integrate multimodal diagnostics. In this case, not only looking for mutations and small nucleotide variations, but also identifying the best methods for clinical diagnostics of structural variation. And often these are DNA and RNA sequencing combined in the clinical laboratory. And I highlight RNA sequencing and fusions and copy number as things that I'm going to highlight in the talk. The methods need to be robust and scalable enough for fairly low quality specimens in the pathology laboratory that are typically FFPE specimens or fresh formalin fixed paraffin embedded specimens. And then we are still trying to figure out in the clinical diagnostic space which tumor and when and what type of methods are best suited in different types of tumors. In other words, genome-wide testing versus focused panels for clinical utility. So I'll focus on structural variation, a specific type of structural variation class, in this case, gene fusions that can arise through different structural variation mechanisms. The most common, I would say the most well understood is chromosomal translocation. And so this is a prototypical example over here of a chromosomal translocation of one gene on chromosome on the left, the other gene on chromosome on the right, leading to a translocation so that two of the genes in green and orange come together in this chromosome on the bottom. And these examples are well known and widely studied and have been known for like two decades and sometimes 3 decades. BCOR ABL1 in leukemias, PML, RARA, ETV6, NTRK3. Classic examples. However, it's important to realize that gene fusions in pediatric cancers or in adult cancers for that matter can also arise from different non-translocation mechanisms. And often these are intrachromosomal events between different portions of the same chromosome. And often they might be cryptic and not detectable by other mechanisms. So one of them is an intrachromosomal deletion, in this case shown over here as a deletion. So on the left is a gene that is colored in red, so we'll call this gene A, and on the right is gene B in blue. And they are all both transcribed in the same direction on the same chromosome. And a deletion can arise by one breakpoint within the red gene and one breakpoint within the blue or the gene B. And that deletion can lead to these two genes that are far away on the chromosome to come together and form a chimeric gene. So this example of this is several known now. I've given 2 examples over here. TMPR SS2 in prostate cancer and DNAJV1 PRKCA in fibrolamellar hepatocellular cancer. The second method that we know, again, intrachromosomal, is an inversion. In this case, the 2 genes are actually having the arrows pointed towards 2 different sides. So they are transcribed in 2 different orientations away from each other on the same chromosome. Now, an inversion mechanism would lead to a change in the orientation such that these genes now become in the same orientation and have portions of the red and portions of blue, leading to, again, a chimeric fusion gene. And so example of this is in NAB2 STAT6. It's in a solitary fibrous tumor, a well-known sarcoma. There are also intrachromosome duplication events that can lead to these fusion genes. And so in this example, you have, again, 2 genes. These are on the same chromosome and in the same orientation. In this case, you have a segment intervening between these 2 genes that gets duplicated or has an extra copy formed. And then that extra copy leads to transposition of these sequences. And now you have 2 genes coming next to each other, which are separated in space by several kilobases. So this is an example of intrachromosomal duplication. And the best example that we know of is this BRAF fusion that is seen in a pediatric tumor called pilocytic astrocytoma. So what are the functional consequences of these? Again, these are a few examples. The classic example that I told you about BCOR ABL1 and other fusions is where gene A and gene B both provide coding sequences to the fusion gene. So in this case, gene A and gene B come together. And by coming together, they are offering coding sequences both from gene A and gene B. So the protein that is made is half gene A and half gene B. So this is a true chimeric protein. Right, so the second example, so this is an example of EWSR1 Fly1. It's a good example of the first type of example. The second category over here is where gene A and gene B forms a fusion gene, but gene A does not provide any coding sequences to the fusion gene. So let's take a look at this. In this case, gene A has an untranslated region. This untranslated region is the only contribution of gene A to the fusion gene. In this case, so there is no coding sequence or no protein coming from gene A, but gene B in this case is being driven by the promoter of gene A. So this example where it's a deregulation of the expression of protein B under the control of gene A promoter is often known as a promoter swap event. And in this case, a great example is myosin heavy chain 9 with USP6 in nodular fasciitis. And then you have other examples where it is not even a true, specifically a gene fusion. What you have is a protein B or gene B is being juxtaposed next to an enhancer element from gene A, where the gene actually is far away from the enhancer. So basically, the enhancer is basically driving this expression of protein B. An example of this would be IgH-MYC. This is a very well-known example in Burkitt lymphoma. So the models of this fusion and how they drive cancer is also falls into mainly two main categories. One of them is called the unitary model, in which you have gene A and gene B fairly constant in terms of being seen only with each other or mostly with each other. And they are also very specific to the tumor types. They are seen together, and they're seen together in the specific tumor context. The other example is modular, where gene A and gene B, whereas gene B is fixed, gene B can have partners with a multitude of gene A's. So you can have gene A type 1, gene A from a different type gene, gene A2, gene A, and so on and so forth. So gene B is, in this case, the effector gene, whereas it can be driven by multiple different types of gene A. And this modular unit is also very well known. And the frequency of this in different tumor types can be variable. And these often we refer to as bucket fusions. ALK is a well-known example of this. NTRK is a well-known example of this, where these kinase fusions drive different tumor types. So how would you do workup for gene fusion? So the classic workup was based on cytogenetics. This is the legacy method that is still used in some context. It has fairly low resolution and is not scalable at all. Whereas you also have FISH-based methods, which also been around for a long time now, has definitely high resolution and sensitivity, and still not scalable, versus the newer age methods that we are now speaking about in terms of genomics, where you use different types of next-generation sequencing methods, either dual anchored or single anchored based methods. And these by far offer the highest resolution, highest sensitivity, and also scalable, if not the most scalable method right now. So in most academic and reference centers, use of different types of panels, which are comprehensive panels, is fairly well established. And some centers might use even more comprehensive, including exomes and genomes for cancers. But for the most part, clinical practice today is mostly utilizing comprehensive panel designs. This is an example of how these panels might look like. There are genes often that are primarily there for the mutation class that we've looked at, genes that are primarily there for the fusion or structural variation class that we looked at, and then genes that are there for both reasons. And so this is an example of a panel that is used in the clinical diagnostics laboratory at our institution. And so I'm going to share for the last 15, 16 minutes or so, I'm going to now share some examples of the utility of applying such broad genomic testing in different types of childhood cancer. So I'll start with some gene fusion examples. So the first example of this is an anaplastic ependymoma, which is a fairly well-known tumor type in children, has been studied in different types of in cancer genome catalog studies. And ependymomas seem to usually have a particular type of fusion, that is C11ORF95 with REL-A. However, in literally hundreds of ependymomas that have been studied, the presence of NTRK fusion has not been described. So here in this case, what you can see is a tumor, and in panel B up top, this radiologic image that you can see, there are some white arrows that are pointing to where the tumor is before the use of NTRK inhibitor. And in panel C, you can see those same white arrows showing essentially dissolution of those white areas that are the tumor foci. You can see at the bottom this pathology image showing the tumor, and to the right of this, these are the NGS or next generation RNA sequencing reads that show a fusion between a gene called CANK1 to NTRK2 or NTRK2. And so this is a relatively unusual finding. First of all, the fusion itself is very rare, described only once or twice. This particular fusion NTRK2 has never been described in ependymoma. However, the presence of this fusion, fairly unexpected, did lead to an attractable target for this patient. And this patient was treated with an NTRK inhibitor in this case. And the treatment did show a very good response, and this patient is actually doing very well right now. This paper is in press right now. I'll show a second example with colleagues at across the street at MD Anderson. This is now a middle-aged man who had a lesion in the spinal area, in the vertebral area. And this lesion, if you see on the right, was a huge lesion that was actually completely encompassing the large blood vessels, leading to an inoperable tumor. So you could not really take the tumor out without damaging potentially some of these vessels. This tumor was then sent to us for clinical testing, and we found a different NTRK fusion in this case called LMNA with NTRK1. And if you see A, B, and then C and D, you can see a distinct change between A and D. For example, in A, you can see all this white space that is being the tumor kind of getting into spaces. Whereas post-therapy in D, you can see that that tumor has fairly dissolved. And this patient got a vertebrectomy done and is doing very well as well. A third example, this is again from our archives, a 16-year-old female with a large mandibular mass. It's a big white tumor that is arising in this radiologic image on the top panel in the left. And you can see some pathologic images showing expression of certain markers that indicate that this is a skeletal muscle tumor or rhabdomyosarcoma. Now, when we did clinical testing for this patient, we found a FUS gene fusion with an TFCP2. This is a fairly new tumor or new fusion that has been described in the literature, mostly in tumors that arise in these bones, in this case, mandible. And it's a fairly rare and totally unexpected fusion in this case. Again, a great diagnostic biomarker and helps with the patient's clinical management. So these are some examples of fusions. I'm going to move on to show you an example of a series of cases where we looked at a different type of alteration, and these would be classified as tandem duplications. Now, tandem duplications by themselves are one of the most difficult alterations that currently exist in terms of clinical genomic analysis pipelines, because they provide a unique challenge in terms of mapping these events to the reference genome, because there are segments that are duplicated and they do not exist on the reference genome. So this is an example of internal tandem duplications in a gene called BCOR or BCL6 co-repressor. This is an old story. We have published this in 2015, along with several other papers coming out from different parts of the world at the same time. This is in clear cell sarcoma of kidney, which is a fairly aggressive renal tumor of childhood. These tumors arise in the kidney and often can not be easily distinguished from a different type of tumor, which called Wilms tumor. And Wilms tumors are often do fairly well, whereas these tumors, if not treated very aggressively, can lead to a very poor outcome. So diagnosing these tumors are very important. However, diagnosing these tumors can be very difficult, because these are fairly rare tumors and not many have experience with these. And B is because these tumors can come in a variety and multitude of different pathologic patterns. So it makes it for a difficult diagnosis for pathologists. In this case, we had one patient that we were studying as part of a large clinical genomics trial. And we found this patient's normal, you can see on the bottom panel where we show some next generation sequencing reads. You can see that in this case, in this patient's normal blood, we found no mutations, but in the patient's tumor, we found a point mutation at the stop codon, it's a stop loss variant in that same gene. So it's a somatic finding. However, stop loss mutations are fairly rare. And if you see and look at this pattern over here, it suggests that there is something called a soft clipping. It's a bioinformatic term highlighting some sort of a processing issue with mapping that is causing these reads to be clipped or partially removed. So this one, this next slide can show you a better image of this. Again, on the top is normal. On the bottom is T for tumor. And you can see on the left that exact same image that I showed you on the previous slide. However, if you can unclip those, so these are soft clipped, meaning these are hidden. If you can unmask these, you can see that all of these actually have some sequences after that were not showing up on the left. You can see on the right. And so when you take those sequences and you map them against the reference genome, you realize that these are tandem duplications in which, if you look over here on the left top, you see that BCOR is a gene that has 15 exons. In the last exon of this gene, you have a segment that is highlighted in green called P, or parental segment. Just below that, I show a parental segment next to the red segment, which is the ITD, or internal tandem duplication. As you can see that the parental segment is basically duplicated in tandem or next to each other. That's why it's called tandem duplication. And so the reads that go from the parental segment into the duplicated segment, these are the reads that get clipped because they are not supposed to have a duplication over here. So once we found that, we looked at all the other clear cell sarcomas in our archives, and we found remarkably that all of these tumors, showing over here in a gel electrophoregram, had an internal tandem duplication regardless of male or female gender. We also mapped out each of these segments. Green, again, at the bottom over here. Green is the parental segment and red is the duplicated segment. And as far as you can see, there is a common segment that is duplicated in each of these tumors, but each of these tumors had different tandem duplications. So given those were very novel findings at that time, now it's a fairly established biomarker and is studied by many groups. We also did some functional studies to identify the effect of these in BCOR expression. You can see on the top that the presence of these duplications or ITDs led to a very high expression of BCOR in these tumors, whereas in similar tumors in the kidney, BCOR expression was very low. Except at the end, you see a BCOR CCN, which is a different type of sarcoma, but in this case, instead of an ITD, it has a BCOR fusion gene, where also the expression might be very high. And on the right, I show some protein expression showing that it's not just the mRNA, but BCOR protein itself is also highly expressed. That led to the use of BCOR antibodies in some of these tumors. And in the middle section, you can see that there are some strong expression of BCOR. Now, we went back to our bioinformatics method of identifying this, and in published data sets, where we did not have the actual samples, but we had published and available data in repositories, we looked through all the clear cell sarcomas that have been sequenced as part of the target study and other studies, and we found the same discordant mate pairs that we had seen in our tumors also show up in these tumors. So that was a remarkable finding. This is a completely independent data set, not sequenced by us, by other groups, and we were able to show that there is a peak near the BCOR exon 15 in every single case that we looked at. So we realized that this was a very classic and defining mutation in this particular tumor, because we did not see this mutation in many other different types of tumors. And then we looked at expression profiling of these tumors and compared it to many other sarcomas and other tumors. And we remarkably found that whether it was a BCOR fusion with CCNB3 or whether it's a BCOR ITD, these tumors all clustered together at the center over here. We can see of this expression profiling. This is a hierarchical clustering, unsupervised, but you can see the CCSKs and the fusion-positive tumors with BCOR all clustered together neatly. So that's a remarkable finding. Now we know, about 5 or 6 years down the line, that many of these tumors that we call undifferentiated sarcomas and other tumors can have both types of alterations defining these tumors. So this table over here at the bottom shows on the first column, you have the different tumor types. The second column shows the incidence of BCOR fusion in these tumors. And then the third column shows the incidence of BCOR ITDs in these tumors. And what we found was that somewhat interchangeably, most of these tumors can have either one. And so this is borne out by the genetic and by the expression studies that I've shown you so far. So changing gears over here for the last few minutes, I'm going to focus on, again, an unexpected finding, but in this case, not of any particular mutation or any particular structural variation, but in broadly of the mutation rate or mutation load in a tumor. So we refer to this as hypermutation. This is one patient from our archive who we published about a couple of years back. This patient presented to our service with what is typically referred to as multiple café au lait spots. And café au lait spots are nothing but a portion of skin discoloration. You can see in figure A and figure B, in panel A and panel B, you can see multiple areas of the back of the head as well as the back of the patient showing dark discolored patches. These are often referred to as café au lait spots. And in a patient with so many café au lait spots, a typical clinical impression is of potentially of a neurofibromatosis or neurofibroma. But because of the presence of multiple of these and the patient presented with a large tumor, which is seen in C and D in the brain or is in the skull, the concern is always for a constitutional mismatch repair deficiency syndrome in which you have biallelic or 2 copies of your mismatch repair genes mutated in the germline, or the patient has inherited these 2 mutations. So in G, H, I, and J, we show that by protein immunostaining for this mismatch repair proteins, we were able to rule out that there was no mismatch repair mutations in these patients. So what could be the cause of this? When we did the next generation sequencing panel for this patient's tumor, what we found was a large number of variants in this patient's tumor. And that would fit with a tumor count or tumor mutation burden of 157, which typically translates to ultra-hypermutated tumor. And so ultra-hypermutated tumor, and I already showed you that there were no mismatch repair mutations. So this typically, by doing mutation signature analysis, can be resolved. And so in our methods of mutation signature analysis, we compared our mutation signature in this tumor to the right to the published mutation signatures on the left, and we found what was a classic signature 10. And signature 10 is typically associated with a germline defect in polymerase mutations, so DNA polymerase gene mutations. And so here we show that we actually did then go in and find a polymerase E mutation in the germline. On the top, you can see these are normal or germline, and in the bottom, you can see tumor. You can see there's a mutation in pol E, the polymerase epsilon that would lead to an alanine at 456 being converted to proline. This is a well-known hypermutator mutation in pol E. And the identification of this led to not only the genomic signature, which led us to do this mutation analysis, but the identification of the tumor mutation burden, mutation signature, and the poly mutation led to multiple clinical management decisions that we made for this patient. This patient is now closely being surveilled for early colorectal cancer, and this patient tumor is considered sensitive to immune checkpoint inhibitor if this patient's tumor does recur. So I hope I was able to show you some examples of unexpected targetable prognostic and diagnostic biomarkers that you can find by broad genomic testing in childhood cancers. I showed you many examples of fusions and other structural variations that can often be sole drivers of these cancers. And of course, there are much more work that remains to be done in childhood cancer, especially in the clinical diagnostic space, including identification of novel targeted therapies and further integrating multimodal genomic sequencing techniques. So with that, I'm going to stop here and acknowledge the large group of people who work in my lab, as well as the numerous collaborators with whom I've worked over the years, some of the centers and sites that have provided all of this information for us. And I appreciate your attention, and I hope you enjoyed this talk, and please feel free to reach out to me if you have any questions, and that will be the end of this talk. Thank you.